Здравствуйте.
Good morning, students.
Good morning.
Good morning. Start our lesson from input control questions.
Okay, good morning.
Answer the questions. First question. Molecular cloning methods. Who answer?
Либо Гульмаров.
Please, Gulmaral. Please answer.
Molecular cloning method. Molecular cloning is a set of experimental methods in molecular biology that are used to assemble recombinant DNA molecules and to direct their replication in host organism. The use of the word cloning refers to the fact that the method involves replication a single molecule to produce a population of cells with identical DNA molecules. Molecular cloning typically uses the DNA sequence of two different organisms, a species that is the source of the cloned DNA and a species that will serve
Okay. Good.
as a living
Next
host
question,
to
please.
Можно я Тулюгазина Алина?
Alina Tuligadina, please.
Restrictases and the function. Restriction endocytosis are a group of enzymes belonging to a class of hydrogases that catalyze under reaction of hydrolysis of nucleic acids. Unlike ecanalysis, restrictases cleave a nucleic acid not from the end of the molecule, but in the middle. In the case, each restrictase recognizes a specific section of DNA with a length of 4 pairs and nucleotides and speeds the nucleotide chain inside or outside the regeneration site. The bacterial genome is protected from its own restriction by methylation of nucleotide residues of adenine and cytosine.
Okay. Excellent, Alina Tuligadina. Next question, who wants answer? Ligases and their action. What is plasmid? Vectors? What are clones? Who wants answer?
Можно Толкинова перезать?
Okay. Толкинова перезать, please.
DNA ligases catalyze the formation of a phosphatase bond between DNA single strands in the duplex form. The covalent linkage of the 5' group of one chain with the 8' 3OH group of another is coupled with a 5-phosphate hydrolysis of the cofactor ATP or NADE. Bacterial DNA ligases hydrolysis of NADE as their energy source. In contrast, ATP is the cofactor for DNA ligases from bacteriophages and eukaryotic cells.
Okay.
The covalent joining of polynucleotides catalyzed by the DNA ligase is a necessary event in a DNA repair. Recombination and most notably DNA replication with rigorous joining of fragments generated from the copying of the minus.
Okay. Good. That next question. Guljanat Orn, please. Please answer.
Plasmida is a small extra former DNA molecule which are cell DNA and can't replicate independently. They are most commonly found as small circular double-stranded DNA molecular in bacteria. However, plasmids are sometimes present in archaea and eukaryotic organisms. In nature, plasmida often carry genes that benefit the survival of the organisms and control selective benign such antibiotic resistance.
Antibiotics. Okay. Good. Next question. Who wants to answer?
May I?
Laura Tokan, please.
Yes. Vectors in molecular cloning, a vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell, where it can be replicated or expressed. And a vector containing foreign DNA is termed recombinant DNA. The four major types of vectors are plasmids, viral vectors, plasmids, and artificial chromosomes. Of these, the most commonly used vectors are plasmids, and the common to all engineered vectors are an origin of replication, a multi-cloning site, and a selectable marker. As a vector itself is generally a DNA sequence that consists of insert and a large sequence that serves as the backbone of the vector. The purpose of a vector is transfer genetic information to another cell and typically to isolate, multiply, or express the insert in the target cell. That's all.
Excellent.
May
Next question, please. Who wants to answer?
Магазила Фарида.
Farida Shumagazila, please.
What are clones? Cloning the immigrants in a natural way or the recipient of several genetically identical organisms that are asexual, including vegetative reproduction or parthenogenesis. The term cloning, in the same sense, is often used in relation to the cells of multicellular organisms. That's all.
Excellent. Next question, please answer.
Можно говорить?
Guldariga, please. Markan
What is
Guldariga.
genome library? Genomic library is a collection of the total genomic DNA from a single organism. The DNA is stored in population of identical vectors, each containing a different insert of DNA. In order to construct genomic libraries, the organism's DNA is extraction from cells and then digested with restriction enzyme to cut the DNA into fragments of such size. The fragments are then inserted into the vector using DNA linkers.
Okay. Very good. Next question, please. Who wants to answer?
Можно я?
Okay.
DNA,
Alina Tuleigazina, да? Yes?
DNA library is a collection of DNA fragments that have been cloned into vectors so that researchers can identify and isolate the DNA fragments that interest them for the study. There are basically two kinds of libraries: genomic DNA and the DNA libraries. Genomic DNA libraries contain large fragments of of DNA in the bacteriages of bacterial derived article chromosomes. The like DNA sequence corresponding to genomic regions that are not expressed, such as introns and 5 and 3 non-cloning.
Okay. Good.
The next question I want to answer.
Please answer.
Yes. About recombinant DNA. Recombinant DNA, molecules of DNA from two different species that are inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. Recombinant DNA technology has three steps. The first is treat foreign DNA and plasmid with restriction enzyme and DNA ligase. In the second step, introduce the recombinant plasmid into cultured plant cells, and in the third step is regenerate new plant from cultured cells. That's all. Thank you.
Excellent.
Thank you.
Continue our lesson. Laboratory work 13. Use of restriction enzymes. Aim of the work studies the role of restriction enzymes, and the plan of the lesson. How do restriction enzymes work? Cutting DNA with restriction enzymes, and watch the video about restriction enzymes. Restriction enzymes. Restriction enzymes belong to a large class of enzymes called nucleases. There are two kinds of exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cut at specific position within the DNA. A restriction enzyme is a protein that recognizes a specific short nucleotide sequence and cuts the DNA only at that specific site, which is known as restriction sites or target sequence. More than 400 restriction enzymes have been isolated from the bacteria that produce them. Like all enzymes, restriction enzymes are highly specific. They cut DNA only within very precise recognition sites or recognition sequence. Many restriction enzymes make strategic cuts at or near the recognition sites, producing ends with a single-stranded overhang. If two DNA molecules have matching ends, they can be joined by the enzyme DNA ligase. In this illustration, you can see the different recognition sequence. The red line cuts the DNA molecule. Red lines, the enzymes cut the DNA molecule. In the first and the second illustration, you can see the enzymes cut DNA molecule and form sticky ends. And the next illustration shows enzymes cut this DNA with forming blunt ends. Enzymes HIA3 and ALU1 cut straight across the double helix, producing blunt end. Cut the DNA across and by producing of blunt ends. The ends of the cut have an overhanging piece of single-stranded DNA. If enzymes cutted the DNA and are forming overhanging strands of DNA, this called sticky ends, because they are able to form base pairs with any DNA molecule that contains the complementary sticky end. Notice that all of these recognition sites are symmetrical, or what is called palindromic. This means that the recognition sequence on one DNA strand reads in the opposite direction on the complementary strand. Cutting DNA with restriction enzymes, microscale quantities. We use very small quantities when working with DNA, so the and the tools are adapted to this micro scale. In the metric system, the prefix micro indicates 1 millionth. It is symbolized by mu, the Greek letter mu. Some examples are 1 milliliter equal 1000 microliter, and the 1 milligram equal to 1000 microgram. You begin the restriction processes by mixing DNA with one or more restriction enzymes in a small plastic microcentrifuge tube. The total volume of the mix is about 20 microliter. Most restriction enzymes reaction are incubated at 37 degrees of Celsius for 1 hour. DNA added the restriction enzymes and are incubated at temperature 37 degrees of Celsius for 1 hour. After incubation, you can analyze the DNA or use it in other kinds of reactions such as bacterial transformation, and we can analyze the separate DNA fragments after the restriction with gel electrophoresis. Control questions. You should answer the questions in next laboratory lesson. And the home task prepares the presentation material, restriction enzymes, and send me by university.
Okay.
Watch the video about restriction enzymes.
Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of these added methyl groups to DNA, while the other cut DNA. The latter was called restriction endonuclease. Restriction enzymes belong to a larger class of enzymes called nucleases. These are of two kinds, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases may cut at specific positions within the DNA. Restriction endonuclease always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as recognition sequence. More than 900 restriction enzymes have been isolated from 230 strains of bacteria, each of which recognize different recognition sequences. Example, restriction enzymes E. coli. E represents the genus name Escherichia. Co represents species name Escherichia coli. These 3 letters are written in italics. R, name of the strain. Roman numbers following the names indicate the order in which the enzymes were isolated from that strain of bacteria. Each restriction endonuclease functions by inspecting the length of DNA sequence and recognizes a specific palindromic nucleotide sequences in DNA. Palindrome means a group of letters that form same words when read both backward and forward. Example, 5' to 3' direction, 3' to 5' direction, 5' GA ATTC, 3 CTTAG, 5 Restriction enzyme cleaves DNA molecules in 2 ways. 1. In one way, they cut both strands of DNA at exactly opposite points to each other. This results in DNA fragments with blunt ends or flush ends, where 2 strands end at the same point. Such cut is generally termed as even cut. 2. But commonly, most enzymes cut the 2 strands of DNA double helix at different locations. Such a cleavage is generally termed as staggered cut. This generates protruding ends. That is, one strand of DNA double helix extends some bases beyond the other. Since the target site is palindromic in nature, the protruding ends generated by such a cleavage have complementary base sequence. As a result, they readily pair with each other, and such ends are called cohesive ends or sticky ends. This stickiness of ends facilitates the action of enzyme DNA ligase. When cut by the same restriction enzyme, the resultant DNA fragments have same kind of sticky ends, and these can be joined together by using DNA ligases. This property of restriction enzymes is of great value for the construction of recombinant DNA. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources. When cut by the same restriction enzymes, the resultant DNA fragments have same kind of sticky ends, and these can be joined together using DNA ligases. The recombinant vector molecule cannot be created unless the vector and source DNA is cut with the same restriction enzyme. Separation and isolation of DNA fragments. The DNA fragments can be separated by gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards a node under electric field through matrix or medium. The matrix is agarose. The DNA fragments separate according to their size through sieving effect produced by agarose gel. The DNA is bright orange colored bands in an ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from agarose gel and extracted from gel piece. This step is known as elution.
Our lesson finished. Thank you for your attention. Good luck, students.
Thank you, and you.
Goodbye.